Hello everyone, you're welcome to this program brought to you by Digital Schools Uganda. We are going to look at biology today, and by the end of this discussion we should have a basic knowledge of biology, what it is, the branches of biology, and then generally why we study biology. So beginning with the word biology, you well know that it's divided in two words. That is bios and logos. These are Greek words, and that is simply the study of life. Logos is a study, and then bios is general life. So we are studying about life, living organisms, all organisms that are living. In the branches of biology, there are so many, several. If we break it down and look at it bits by bits, you must have heard of botany. That is the study of the plants. You must have heard of zoology. Zoology is the study of animals. Anatomy, the study of the structures of the body. We have nutrition. That is the study of feeding. We have mycology, the study of fungi. We have ecology, the study of interactions, how living organisms are interacting with their environment and with one another. Entomology, the study of insects. We have physiology, the study of how bodies function, the study of how our bodies function. We have genetics, that is the study of heredity. Okay? Then we have other branches like taxonomy, which we are going to look at it in details. Taxonomy, this is a very important branch of biology. We're going to look at ornithology, the study of birds. People like birds, you see birds around. Ichthyology, the study of fish. So generally, why do we study biology? That is a very important question that each of you should be asking yourselves. Why are we studying biology? So just a few, adding to what you already have in your head of why you study biology. Of course, we get knowledge. Knowledge. This knowledge can help us treat the sick. Doctors actually study biology. Teachers, we study biology for a reason. So the main thing is we want to know how our body functions. You should know that biology is the study of life, so we need to know how our body functions. We should know how get the knowledge to conserve the environment. We should the knowledge of manufacturing drugs are from plants. Most of the drugs are manufactured from plants, so we also get that knowledge from biology. How do you treat the sick? When you fall sick, the doctors treat you. It is from biology. So when you follow up biology, you'll end up being those. You'll be nurses, you'll be doctors, you'll be pharmacists, because it's an important branch of science. So we are studying living things. That means we need to know the characteristics of living things. We cannot look at living things without knowing the characteristics of these living things. What are some of the characteristics of living things? So this year we are looking at, of course, we can either have, mostly you see plants, and then you see animals. Like for us, we are animals. And you know very well that we depend on plants. We're going to look at functions or characteristics that make just living things in general, and then you also look at the characteristics that make plants different from animals. So one of the characteristics is nutrition or feeding. We have nutrition, and this nutrition is feeding. Every organism has to feed, has to eat, so that it can undergo its metabolic processes. Okay? You eat so that you can live, to get energy to drive processes of the body. Like we're looking at physiology. Those processes need energy, and the energy is from food. Then respiration. If you're going to eat, that food is supposed to be broken down to provide the energy, and that's where respiration comes in place. nutrition, the food is in the body, respiration, the food is broken down to release the energy that you need. We have excretion. Now, if you ingested food, there is part of that food that will not be digested by the body, and so it is a waste. And every process that is taking place in the body, every metabolic process, releases some toxins or some waste products. It has to be eliminated from the body, and that is excretion. We have reproduction. If you're going to be living, you must have continuity. If you die, there should be continuity of other organisms, and that's why reproduction comes in place. Every living organism has to reproduce. If it was not that way, we will probably be living for thousands of years. But because of reproduction, we only live for a short time, and then we leave our young ones to continue. Then we have movement. Movement is also important. Living organisms move. Most of the times, people confuse movement and locomotion. People always confuse that. Movement, every living organism, the plants move and the animals also move. We have growth. Why would you not grow when you're eating? When you're respiring and all this, you have to grow. From a young one, an adult, produce and probably die, and like that. So growth is also an important part. How are you going to be responding to your environment? You need to be sensing the changes in the environment. We call it stimuli. So that's where an important characteristics called sensitivity. Sensitivity. Okay? Sensitivity or irritability comes in. Things in the environment always irritate us in either positively or negatively. So that is irritability. Now, these are characteristics that are common to both plants and animals. But then there are some differences that exist in these organisms, the plants and the animals. Let's look at the differences. How do we tell that this one is a plant and this is a characteristic of an animal? Say they list down characteristics, and you need to know if that organism is a plant or an animal. There are differences that exist between the plants and the animals. And that one, we are just going to mention them, we are not going to write them. But with this table, if you're making any difference in biology, you need to have a table so that it makes everything easy. So every plant cell has a cell wall. All plants have cell walls in their cells. The animals do not have the cell wall. These ones, the plants, since they are autotrophic, since they make their own food, they have chlorophyll, and then the animals do not have that chlorophyll simply because they don't make their own food. They depend on food that is already made. Then the response to stimuli in plants is very slow. And that one for animals, the response to stimuli is very fast for survival reasons. In plants, growth occurs throughout life. A plant will always grow. Every single day, every single year, a plant will always grow until you cut it. But an animal reaches some point and it dies. Okay? In plants, growth occurs at particular parts. Always, usually, the tips. We call them the meristems. But in animals, growth occurs all over the body. Your arms grow, your feet grow, your belly grows, every part grows. But for plants, it is always the tips that grow, so that is important. We are talking about study of life. Biology being the study of life. We have seen living organisms. We have differentiated them into plants, and we have also differentiated them into animals. How do we study these living organisms? We need some kind of instruments that we have to put in place to help us study these organisms. Because some of them are so small that you cannot look at them and easily identify. For us, animals and plants, we can see, yes, this is a person, that is a goat. But there are some organisms that are very small that needs some instruments that can help us identify them. And usually we use the hand lens and we use the microscope. Now the hand lens is used to magnify relatively larger objects. They are already big, you can see them with your eyes, but looking at them clearly to identify more structures. Say you are looking at a small insect and you need to see the antenna clearly, or the hairy body that it has, you have to use a hand lens. And then you have a microscope. Now, when you look at the magnification of a hand lens, it is always a smaller magnification, like 10. So we have the hand lens and then we have the microscope. The hand lens is pretty simple. You will see it in that diagram down there. Then the microscopes, these are more sophisticated than the hand lenses. We have basically two kinds of this. We have the electron microscope, and then we have the compound light microscope. Those are basically the two. Now, the electron microscope uses the beam of electrons. This is more powerful, and usually we use dead specimens on them. But the one which you usually use in schools and in simple labs, compound light microscopes. This one simply uses light to magnify the organism. The structure of the microscope is clear, as you can see it there. We are just going to look at the function of each part, as you see it labeled in the diagram. And one of it is the eyepiece. You can see in the eyepiece, this is just enabling us, like the name says, eyepiece. That's where you put your eyes to view a specimen. We have a barrel that is the body that holds the... So like we said, we have the light microscope and then we have the electron microscope. The electron microscope usually uses a beam of electrons to magnify objects and usually uses dead specimens. The light microscope is a common one that we use in laboratories, in schools for example, and then it basically has parts that we need to look at each of the functions as stated right here. We have the eyepiece. This is the part we use for viewing a specimen, where you put your eyes, as the name says, eyepiece. We have the barrel, that is the one that holds the eyepiece in position. We have the nosepiece, that one is the one which holds the objectives. We have the stage, where you're going to put your sample. We have the mirror that reflects the light from a source into the specimen. We have a base or a stand, that is the one that enables the microscope to rest on a table. We have a diaphragm, and then we have a condenser, that is a system that concentrates the light as it enters to the base where the specimen is. And then we have the arm, you use it for holding the microscope. According to the image that you see on your screen, you see we have a coarse adjustment knob and we have a fine adjustment knob. That one, the coarse, like it says coarse, it magnifies, it adjusts the microscope more widely, and then the fine adjustment tries to focus it very minutely so that you can see a detail. And then another thing, maybe the objective lenses. Usually you have different objective lenses, the low power, the medium power, and the high power. So depending on each of those, they have different magnifications. If you want to use maybe to view something on a smaller magnification, you use the low resolving power. If you want to look at it at maybe a higher magnification, use the higher magnification objective lens. All these are always multiplied by the eyepiece magnification to get a total magnification. The microscopes are very delicate and expensive instruments that have to be taken care of properly in the laboratory. And just a recap of a few, you know that you have to hold it with both hands. It should not drop at any time when you're carrying it. And then it should never be dropped. For sure, you drop a microscope down and then it breaks, and you have said it is an expensive apparatus. It should always be in an upright position. A flat table and then placed in an upright position. Always wipe the lens. If you are cleaning the lens, you use a soft cloth. a soft cotton cloth. Don't get something rough and then you're using it for cleaning the microscope. You can harm the lens and you'll not have proper functioning. And then it should also be kept in a special designed box. Anytime you go to see a microscope, you'll discover that there is a casing that it is kept into so that it can protect it from both dust and injury in case it falls. Determination of the magnification of a microscope. If you're looking at the magnification of a microscope, we always have the eyepiece and then you have the objective lenses. So at any time, the magnification of the microscope will be multiplying the eyepiece magnification times the magnification of the objective lenses. Usually you will see them labeled. If the eyepiece is magnification times 10 and the objective lens is the magnification, let's say, times 40, the total magnification of that microscope will always be 10 times 40. So it will be a 400 magnification power. And so is if the magnification of the objective is, say, 60, that is all the process that you have to do. As we have looked at life being the main thing that you're studying in biology, and we have seen how we study these life forms, it is important now that we have instruments like the microscope, we look at the cell.